Now look at this.
What's up, penguins? Today we're going to do topic 3.5 on photosynthesis. Photosynthesis takes place in the chloroplast, and there's two main reactions. You have your light reactions that take place in the thylakoid membrane, and you have your Calvin cycle that takes place in the stroma. The light reactions produces the ATP and the NADPH that's needed for the Calvin cycle. So in the thylakoid membrane, you're going to see there's two different photosystems. So right here we have photosystem 2, and here we have photosystem 1. So both photosystems are going to have a chlorophyll molecule in their reaction center complex. And so you see how we all have all these like pigments on the outside? Those are your antenna pigments, and they're going to absorb the light energy, and they're going to kind of funnel all the light energy into that reaction center complex. Once it reaches that reaction center complex, there's going to be a primary electron acceptor that's going to take the electron and it's going to send it down the electron transport chain. In photosystem 2, we're going to see a process called photolysis. Photolysis is where you're going to use light energy in order to split a water molecule. When you split that water molecule, you can regenerate the electrons that have been lost by photosystem 2 when the primary electron acceptor took the electron from our chlorophyll molecule. So there's two different types of electron flow. So this you see right here is a linear electron flow. So we're going to have the electron moving from photosystem 2 through an electron transport chain to photosystem 1 through an electron transport chain, and then finally going to NADPH. Versus cyclic electron flow is just going to deal with photosystem 1, and then it rotates past that electron transport chain that's between photosystem 2 and photosystem 1. So the reason why we have two different ways that the electrons can flow is that linear electron flow is going to make a 1-to-1 ratio of ATP to NADPH, versus the cyclic electron flow is going to make just ATP. So let's talk about the electron transport chain that takes place between photosystem 2 and photosystem 1. So every time that the electron drops in energy, it's going to pump a proton against the concentration gradient. So what's going to happen is that those protons are going to get pumped into the thylakoid space. So they get pumped into that small space. So if you remember about pH, that's going to cause the pH inside of the thylakoid to decrease because it's going to have a high concentration of hydrogen ions. Okay, that concentration of hydrogen ions is then going to fuel this ATP synthase. This is an enzyme that's going to synthesize ATP. So it uses the concentration gradient. As it falls down that gradient, it releases that potential energy that's then going to drive ATP synthase to synthesize the ATP. So here we see the Calvin cycle. In the Calvin cycle, we're going to have 3 molecules of carbon dioxide is going to bind to RUBP using the enzyme Rubisca. We're then going to reduce this carbon by donating the electrons that we found from the light reaction using NADPH and 6 molecules of ATP. That's going to make something called G3B that's going to leave. The 5 molecules of GTP are then going to return to be able to rearrange back into RUBP, which uses 3 molecules of ATP. If you've been paying with just success, bye y'all.